Hey guys, it's me, Mr. Plazak. Today what I'm going to talk a little bit about is probably one of the more complex ideas when we talk about the muscular system, and this is the sliding filament theory of muscle contraction. I know it sounds like a very scary group of terminology, the sliding filament theory of muscle contraction, but basically it's just going to explain how a muscle is able to contract. So I know this is a really complex and abstract idea, but I think as you watch and you progress through this video, you're going to have a great, better understanding of how the muscle itself is able to go through the contraction process. So I'll break it down step by step in a manner that will be a little bit easier for you to be able to grasp. So when we take a look at an actual muscle cell itself, we also give the terminology when we take a look at skeletal muscle. We call individual muscle cells, we call them muscle fibers. And for all intents and purposes in the land of make-believe, let's consider this particular structure right here. I know it's spaghetti, but we're going to consider this spaghetti-like structure right here. This is going to be a muscle fiber. So when you take a look at this individual muscle fiber itself, this is what we're going to, when we make up actual skeletal muscle, we take a whole bunch of these fibers and we basically join them together. So this is this individual muscle fiber that we have here that you can see, okay? When we take a look at the membrane that's surrounding this thing, okay? This muscle cell has a particular membrane that's surrounding it, and we call this membrane the sarcolemma. So this membrane that is surrounding this individual muscle cell is called the sarcolemma. So this is the sarcolemma, this membrane that's actually surrounding this particular muscle cell itself. So then surrounding this sarcolemma, you have another connective tissue membrane that's going to be wrapping around this. This connective tissue membrane that's going to be wrapping around the sarcolemma is called the endomysium. So this has another membrane that surrounds it as well that's called, or a connective tissue wrapping that's surrounding this as well, that's called the endomysium. Now, when I take a look at this individual muscle fiber itself, if this were going to have to move my arm or something like that, there's no way possible that this little individual muscle cell or muscle fiber alone, this skeletal muscle fiber alone, could possibly move my particular bone that it needs to move. So what does this little tiny muscle fiber have to do? This little tiny muscle fiber is going to have to join with another muscle fiber. So basically it's going to have to find another muscle fiber and it's going to have to join with it. So we're going to take these two and we're going to join them together, and then these two are going to join with another one and another one and another one and another one and another one, and we're going to have to wrap them up. And we're going to wrap these individual muscle fibers all together in what we call a fascicle. So this is an actual fascicle itself that you can see here, and this fascicle that I have here, is wrapped in another membrane. The membrane that this fascicle is wrapped in is called the perimysium. So you can see here, this is the perimysium wrapping all these individual muscle fibers together, and it's wrapped in a membrane called a perimysium. Now this is a fascicle. This still alone, okay, when this whole entire unit of these muscle fibers, when they all contract, this still alone cannot get a bone to be able to pull on it enough to give it enough force to be able to move that particular bone itself. So what does this fascicle have to do? Well, this fascicle is going to have to join with another fascicle that we have here. And we're going to join these fascicles all together. Let me grab some saran wrap, and I'm going to join all these fascicles together. So now I took all these fascicles and I joined them together. So now all these fascicles are going to be joined together, and these are going to be joined together by another connective tissue membrane that we call the epimysium. And the epimysium then at the end is going to run continuous with what we call tendons. So these are going to be tendons down here, and these tendons are what's going to actually attach to the actual bone itself. Now this whole structure here is a skeletal muscle. This whole structure here is a skeletal muscle that I have with these tendons on the end, and then right here we have all these muscle fibers that are joined together, or all these fascicles that are joined together, and this is where the muscle contraction, when this occurs, then it's going to generate enough force to be able to cause bone to actually move. Now remember, skeletal muscle pulls on bone. So this whole structure is going to pull on bone. When these muscle fibers contract, they get shorter. So when you contract, you get shorter, and this structure is going to actually end up pulling on actual bone itself. So when you take a look at this image, this is an image that we just kind of went over earlier with these different connective tissue wrappings here. You can see here we started out right here with the individual muscle fiber. This is also called a muscle cell or a skeletal muscle cell. Remember, it has a membrane around it that's called the sarcolemma. Okay, this sarcolemma then is surrounded by what we call the endomysium. And remember now that this individual muscle fiber alone can't nearly contract with enough force to be able to move a particular bone. So this individual muscle fiber has to find another muscle fiber to join with, another muscle fiber to join with, another muscle fiber to join with. And they join with a whole bunch of muscle fibers, and we wrap them in a membrane, okay, that we call the perimysium. This structure is called a fascicle. So you can see this is a fascicle that's right here. That's basically a union of a whole bunch of muscle fibers themselves all together, and they're wrapped in a membrane that we call the actual perimysium. So this is the perimysium, this connective tissue wrapping that's surrounding the individual fascicle itself. Now this individual fascicle, when it contracts all together, it alone can't generate enough force in order to be able to move an actual bone itself. So this fascicle has to join with another fascicle, and this fascicle has to join with another fascicle, and this fascicle has to join with another fascicle, and this fascicle has to join with another fascicle. And then we join and we unite all these fascicles together, and we wrap them together in a connective tissue wrapping that we call the epimysium. And then you can see the epimysium at the bottom then runs continuous with what we call a tendon, and then the tendon is going to actually attach the bone. This whole entire structure is a skeletal muscle. So this could be the semitendinosus or the semimembranosus. This could be your trapezius, the orbicularis oculi, but this is going to represent skeletal muscle. And for the most part, guys, skeletal muscle is going to move bone, is its major job. One of its functions is it's going to actually move the actual bone itself. This is going over all those different skeletal muscle attachment points that we have, the epimysium. This is what a tendon is. This is another structure we call the aponeurosis that we can take a look at there. But the main goal, guys, the main goal of what I'm going to talk about today is to just kind of get over the gist of how a muscle is able to contract. So I'm going to move into now this whole entire idea of how we're able to get a skeletal muscle to be able to contract. So I'm going to take that individual muscle fiber and I'm going to break it down even further and further and further till we get to a single unit of contraction in that actual muscle fiber itself. So we take a look at the major functions of actual skeletal muscle. So the first movement, the first function that we have is it's going to produce movement. So this is what's going to be able to produce movement in your body is the actual contraction of the skeletal muscle. It's going to a lot of times pull on this bone. It also helps maintain posture, so it keeps you upright. These muscles are going to have that ability to keep you upright, position and maintaining your posture itself. It's also going to stabilize joints. When you take a look at like your knee joint itself, that synovial joint, that hinge joint that you have that's there is all stabilized by muscles that are surrounding it with those tendons that are associated with it, or keeping it firmly in shape and preventing it from becoming really loose and then really unstable. Also, when we move muscle, it's going to generate heat. Anytime you have that intense amount of exercise that you do, you're generating heat. And that heat then is going to help warm up your actual body itself. So these are the four major functions that we have with skeletal muscle itself. When I take a look at the microscopic anatomy of the skeletal muscle, remember the membrane that surrounds the skeletal muscle, this individual muscle fiber itself, okay, is called the sarcolemma. If I take this sarcolemma, okay, and then I begin to take a look inside the sarcolemma and I peel it back even further and I look inside this individual muscle fiber, I'm going to see a whole bunch of structures in this muscle fiber once I break it down that are called myofibrils. And inside this individual muscle fiber as well, I'm going to find a whole bunch of unique organelles that are associated with the skeletal muscle. One of the unique organelles that are associated with the skeletal muscle is called the sarcoplasmic reticulum. It's just like a smooth endoplasmic reticulum that you learned about in biology, but it has the ability to hold on to calcium. And calcium is a really important thing that's needed in order for a muscle to be able to actually contract itself. So you can see in this picture that's right here, okay, we have an individual muscle fiber. All right, this is this muscle fiber that we talked a little bit about earlier that I had with that spaghetti strand that's right here. Now I'm really breaking down this individual muscle fiber itself. You can see here is the membrane that's surrounding this muscle fiber. This membrane is called the sarcolemma. You can also see in here, when I take a look inside the sarcolemma and I peel it back, I can see a whole bunch of these other smaller like units that we call, these smaller like units that we have here are called myofibrils. So there's a whole bunch of these myofibrils that are in here as well. Inside here, we're also going to find that sarcoplasmic reticulum. You can see that also the skeletal muscle is multinucleic. It has a ton of nuclei that are associated with it as well. You also see that there's a banding pattern that's associated with the way that these myofibrils are set up. They have dark bands and they have light bands. So I can see when I look at the outside of this structure, I can see there's some dark bands, there's some light bands. Like right here is a dark band. Beside that, there's a light band, a dark band, a light band, a dark band, a light band, a dark band, a light band. These dark and light bands, we give them a name. A dark band is called an A band. A light band is called an I band. If you look at the second letter in the word dark, it's A. So we have an A band. The second letter in the word light is I. So we have a dark band and we have a light band. So dark and light band are the two types of banding patterns that we see when we take a look at this actual skeletal muscle itself. Remember, the job of this particular lecture that I'm going over right now, guys, is to explain to you how an individual muscle cell itself, a muscle fiber itself, has the ability to contract. So we're going to take a look at this whole entire unit here, and we're going to break this down even further and further until we get to a little tiny contractile unit, and we're going to show how this little unit here has the ability to contract, and then that's going to cause the whole entire skeletal muscle to have the ability to contract itself. Here's what I'm talking about, the I bands. Remember, the I bands are light bands. And they're light bands because there's less stuff that's associated with them that gives them a lighter light color. A bands, they tend to have more stuff that's associated with them that tends to give them a lot darker color. So that's the I bands and the A bands, and we take a look at the microscopic anatomy of the actual skeletal muscle. Okay. This is where things get a little bit confusing here. So all I've done is I've come back, if I go back to this very thing that's right here, and we take a look at this myofibril. So I'm going to take this myofibril that's right here, and I'm going to blow it up so it looks a lot bigger, okay, in a bigger picture that's right here. So this is going to be this myofibril right here. I've now blown up this myofibril. And this is what the myofibril looks like when I actually blow it up. And I can see when I take a look at this myofibril, I see I have an I band that's right here, that's a light band. And when I look at this light band, I see that there's not a lot of stuff associated with it. There's just a little bit of these blue lines that are associated with them. And these thin blue lines that I have that are in this image that's right here are called actin. When I take a look at an A band, I see that an A band has a lot of different type of these little structures that are associated with it. So you have myosin, and you have actin, okay? Myosin are these little tiny red bands that are in here. The actin is the blue. When I look at an A band, it has both myosin and it has actin in it, okay, for the most part. So for the most part, when I look at an A band, it has both myosin, which is in the red, and then it has these little blue-like fibers that we call actin. So there's myosin, there's actin, so there's a lot more stuff here, which is going to give it a darker appearance, which is why we call it an A band. Now, if I take an I band, an A band, and an I band, and I join them together, I'm going to have a contractile unit of a muscle that we call a sarcomere. So now what I'm going to do next is I'm going to join these bands together in the upcoming here, and we're really going to blow this unit up, this right here. Now, there is a little area in the actual middle of the actual A band that we call the H zone. And the H zone itself doesn't contain as many fibers. If you notice in this H zone, I don't see the actual actin that's in there. It's just strictly myosin that's kind of located in this A zone, assuming that the muscle's not contracting at the actual moment itself. So when I take a look at the sarcomere itself, okay, a sarcomere is a contractile unit of a muscle. A sarcomere, when I look at this picture that's right here, is the I band, the A band, and the I band. Just this little unit right here is a contractile unit of a muscle. So I just took this myofibril from way back here, and I broke it down even smaller so it looked like this, okay? And now I just took an I band, an A band, and an I band, okay? And this little unit right here is an individual contractile unit of a muscle, and we call that unit a sarcomere. And a sarcomere is composed of these little tiny myofilaments. You have very thick filaments, okay, that are called myosin filaments, and then you have thin filaments that are called actin filaments. So thick filaments, they tend to be a little bit thicker, they're called myosin, and then thin filaments, they're a little bit thinner, they're called actin. Actin was represented in blue in that previous diagram. The myosin was those dark red bands that we saw in there. Thick filaments, like I said, are composed of this myosin. It has a protein that we call myosin that's in there. So this is a protein that's called myosin. Note, bodybuilders love to eat protein. Why do they love to take in protein? Because protein is a major component of the actual development of the actual muscle itself. It has ATPase enzymes. So it has these enzymes that are associated with it. They're going to be associated with the ATP molecule. Also, when I take a look at these thick filaments, they have these little tiny heads or extensions that we call myosin filaments, or we call them myosin heads. Is one of the things that we're going to notice that's very characteristic of the actual myosin filaments themselves. They do overlap actin just a little bit. Those areas that they overlap that actin somewhat tend to be those areas where we see those A bands a lot of those times. Those dark bands is where we tend to have an overlap, minus then that one point we have that M line that's there. Thin filaments are actin filaments. They're composed of a protein that's called actin. They're anchored in place by something that we call a Z-disc. So I'm going to take this picture and I'm just going to blow it up even more, where we're just taking a look at an individual sarcomere right here. So this is the sarcomere that's right here. You can see here's the myosin here that's in red, and you can see these little tiny heads that are on the actual myosin itself. These are called the myosin heads that are here. Here's your actin that's sitting right here. Your actin's in this actual blue itself. So you have actin, then you have your myosin, you have your actin, myosin. As we move down this particular line that's here, you can see that. You can see this actin's anchored in place in this little area that we call the actual Z-disc. What's basically going to happen, guys, when this muscle contracts is this actin is going to move in this M-line area. It's basically what it's going to do. So right now you have something that looks like this, but basically what's going to happen is it's going to move in. It's going to look something like this. So you have this. It's going to move in like that. So you're just moving into that open space is what these actin molecules are basically going to do. My fingers are going to represent the actual actin in what I'm showing here. So this actin is going to go like this, and it's going to go in like this. Okay, what's going to move, what's going to cause these actin molecules to move in is the myosin heads are going to latch on to the actual actin and cause it to move in an inward-like fashion, is what's going to happen when a muscle actually goes about contracting. So at rest, like I said, there's a bare zone that lacks any actin filaments. It's called the H zone. Prior to all of this, in order to get a muscle to contract, you have to have calcium has to be able to be released in order for a muscle to be able to contract. And I'm going to go over step by step how the muscle has the ability to go through this actual contraction process. But remember, the sarcoplasmic reticulums, just like the endoplasmic reticulum that you talked about in your regular general cell biology class, your sophomore year, and this one's specialized because it stores and releases calcium, is what it basically does. This is another picture that I have right here of the actual myosin and the actin. This gives a good depiction, guys, of the actual myosin heads that are located right here with the actual actin itself. And you can see this middle area here we have is called the actual bare zone. And this actin, or the H zone, this actin is going to move in this area. These myosin heads are going to latch on to that particular molecule itself. So this image here is just kind of a blown up image of an actual actin molecule. And the reason that I wanted to show you this is there's two other protein or two other filaments that are associated with the actual actin itself. One is called troponin, and you can see troponin is kind of this blue ribbon that's wrapping around the actual actin itself. Now these actin molecules have these active sites that are on them, and these active sites is what the actual myosin heads are going to bind onto. So these myosin heads are going to bind on to these particular active sites that are here. Okay, and then they're going to cause the actual actin to move into that H zone area that we talked about. In order for these myosin heads to be able to latch on to these active sites, we have to get rid of this troponin. We have to get rid of this tropomyosin. So this tropomyosin is covering up these active sites that are on the actual actin itself, and we have to move these out of the way so that these myosin heads can bind onto those active sites to then latch on and then move this actual actin in an inward-like fashion. What the tropomyosin is held in place by is a molecule called troponin. So troponin is kind of acting like a clothespin that's holding on the tropomyosin to the actual actin itself. Now, what causes the release of the tropomyosin is calcium. So calcium, when it binds onto the troponin, it's going to cause the unraveling a little bit of the tropomyosin to expose these active sites, so then the actual myosin heads can actually latch on. So basically these myosin heads are then going to have the ability to latch on. These myosin heads then are powered by ATP. So in order for these myosin heads to work, they basically cock themselves back, they lock onto the active site, and they move the actual myosin heads in an inward-like fashion, move the actual actin in an inward-like fashion towards that actual H zone is what they're going to do. What releases the calcium? The sarcoplasmic reticulum. So the sarcoplasmic reticulum is going to release this actual calcium itself. In order for this calcium to be released, you have to have a neurotransmitter, okay, or you have to have a neuron, and a neuron is going to fire its neurotransmitter onto the surface of the sarcolemma. So when that neurotransmitter is released onto the surface of the on the sarcolemma, on the outside of the muscle fiber. So you basically have, if this is going to be a representation right here, where my hand up here, this is representing the end of an actual neuron itself. Okay, this is the end of the neuron itself at the axon terminals. Here in this area here is where you're going to have your neurotransmitter that's going to be released. So a neurotransmitter is released onto the surface of the sarcolemma. Okay, this is a muscle cell right here. My individual muscle fiber, this right here, is represented by my arm. So I release this onto the surface, this right here. When I release this onto the surface, it's going to stimulate the muscle to take in more sodium. Okay, the name of this neurotransmitter is acetylcholine, or ACTH. So this ACTH then is going to stimulate the muscle then to take in more sodium. When you have an influx of sodium that rushes into the actual muscle cell itself, that's going to then stimulate the actual sarcoplasmic reticulum to release this calcium. This calcium is then going to bind on to the troponin. So the calcium binds on to the troponin, causing the tropomyosin to unravel, revealing these actual active sites themselves. And now that we are able to have these active sites exposed, these myosin heads can now latch on and move the actual actin in an inward-like fashion into this actual H zone itself. So this is kind of how a muscle has the ability to contract. Remember, ATP has got to be present in order to get these actual myosin heads to latch off of the active sites and then cock and reload and then go on to the next active site and then keep pushing this in that inward-like fashion towards the actual H zone itself. So you can see this is just kind of a generalized diagram right here of in a relaxed muscle cell. Okay, this is dealing with, you can see up here, you have this protein complex that's up here. This is representing the actin that's up here. But you can see that you'd have the troponin and the tropomyosin that's covering up these active sites. So they're not going to give the ability of this myosin head to actually latch on, that is here. And basically you have the calcium that's going to be released, and when that calcium is released, okay, that calcium is released because we had a neurotransmitter or a neuron that fired into that actual muscle and released that neurotransmitter, which then stimulated the actual muscle itself to take in more sodium. That influx of sodium caused the sarcoplasmic reticulum to release calcium. That calcium then is going to bind on, okay, the troponin, unraveling the tropomyosin, and now I can see I have these active sites that are present here. These active sites now give the ability of this myosin head to latch onto it. Now when you take a look at these actual myosin heads, when they latch onto the actual actin, what this structure here is called, it's called a cross bridge. So this is representing an actual cross bridge itself, this complex of the myosin head latching on to the actual actin. If it's like a bridge, because see, if this was a bridge, I can walk all the way across this bridge and I can make my way to the actual actin itself. What's basically going to happen then is this myosin head is going to unlatch. Okay, in order for it to unlatch, ATP is required, and it's going to bind on to the next active site, moving it in. In this case, this one's moving it in. And this one here, this one will latch onto this one, and it's going to pull it in this way. These guys are latching onto the next one here, and they're pulling it in this way. This is that H zone, and the actual muscle itself, this actin, is going to actually move. So as you can see, we're moving in this inward-like fashion, and that's kind of the sliding filament theory of muscle contraction when we take a look at it. The next thing I'm going to show you guys is kind of a video that's going to explain the whole entire process, and I'll stop and I'll pause this video as I go to kind of help you with the actual understanding of what's going on in the process. So you can see, guys, here we have our actual skeletal muscle itself. So this is our skeletal muscle that we're taking a look at itself. And the skeletal muscle is going to power us to do a whole bunch of variety of different things, from walking to riding a bike. When I break down this skeletal muscle, this individual muscle fiber itself, okay? So when I break down an individual muscle fiber itself, I see these structures that are in here, and these are these structures that we call. So you can see, guys, when you take a look at this image that's right here, you can see here's our actual sarcomere that we have right here. Here's my myosin, here's my actin. Here's my actin on this side, and these are the myosin heads that are right here. And basically, this myosin head is going to latch on and then move this actin in an actual inward-like fashion when we take. We're going to take a look here. As you can see right here, guys, is the actual actin itself. So this is the actin that's right here that's just lighting up right now, that's blue. This is the tropomyosin, and then the yellow is the troponin. So this is the calcium that's being released. So calcium is going to be released. When calcium is released by the sarcoplasmic reticulum, it binds on the troponin. That unraveled the actual tropomyosin, exposing the active sites. Now that the active sites are exposed, these myosin heads that are right here can latch on to the actual active sites, and they can pull in this actual actin itself. Remember, these myosin heads that are sitting right here, are powered by ATP. In order to get this myosin head off, ATP actually has to be present. So these are going to continually go through ATP, moving this actin towards that actual H zone itself. And this is the actual muscle contraction that we talk about with the sliding filament theory of muscle contraction. When the calcium goes away, the muscle will then relax. So we'll watch this in action time and time and time again as this is going to latch on. Remember, it latches on, that myosin latches on, it forms a cross bridge, and this is going to continually bring in that actual actin itself. The muscle's relaxing, calcium is leaving, and this will happen time and time and time again during actual muscle contraction. Remember, if ATP is needed to be present in order for that muscle to be able to for that myosin head to be able to latch on. This is the myosin head. In order for it to be able to actually get off and then latch on the next active site and move it in that inward-like fashion. If ATP is not present, this myosin head can't get off of the actual actin itself. And this is something that you have in life that's called rigor mortis. Right after an organism dies or an animal dies or you die, you basically lock up. And you have that time when you're actually locked up. And the reason that you're locked up is because that muscle no longer has ATP that's present in it, and these myosin heads can't get off of that particular area. This lasts for a given period of time in the actual organism itself, and then basically these proteins begin to degrade and the muscle loosens up and you get out of this rigor mortis state. I hope you enjoyed learning a little bit about the sliding filament theory of muscle contraction. I know it's a very complex idea, but if you go back through this video again and again and again, eventually you'll grasp on to one of the more complex ideas in the actual muscle system, and actually one of the more complex ideas in dealing with human anatomy and physiology is this whole entire, how does a muscle have the ability to contract? And hopefully I broke it down to you in a little bit more easier words for you to be able to understand. I know when I was a high school student taking anatomy physiology, I really didn't understand this concept, and I really didn't fully grasp the concept until I went on to actual college itself. I know this is a very difficult idea to grasp, but like I said, just take your time. You can pause this video and write things down as I went over it. I wish you the best of luck in preparing for the muscle test. Like I said, if you have any questions about the sliding filament theory of muscle contraction, don't hesitate to ask.